A very good afternoon to all the participants and welcome Dr. Chandrasekhar to this second deliberation of our FDP. As we all know that this FDP has been basically addressed to the applications of CRISPR-Cas gene editing tool. And Dr. Chandrasekhar, you know that this is an AICTE initiative, and this is the full, the sponsor is AICTE, and the entire academy is called the AICTE Training and Learning Academy, ATAL. So we are very, very happy to have you here today. And I'm very proud that you have accepted our invitation, kind invitation. In the morning, we already had our inauguration and Dr. Shobik Maiti deliberating on his work. And now we will move on to your lecture to the audience. And the audience is comprised of faculties. There are research scholars. There are postgraduate students, and also from other factions also we have trainees also. So to the audience, I would like to introduce our next speaker, Dr. P. Chandrasekhar. So Dr. Chandrasekhar is the principal scientist in the Stem Cell and Developmental Biology Group in the Center for Cellular and Molecular Biology, Hyderabad, which is a CSIR lab, also in short known as CCMB. All of you probably know CCMB. So Dr. Chandrasekhar, he obtained his PhD from CCMB itself, and then he worked in the areas of stem cell biology and mouse genetics. And during his postdoctoral research at Mount Sinai Medical School in New York, in Stem Cell Bangalore, and in the University of Edinburgh, he focused on understanding the auto-regulation of nanog and studying the novel pluripotency factors pathways in the murine embryonic stem cells. Dr. Chandrasekhar was a Wellcome Trust DBT Alliance Early Career Fellow and also at the STEM Bangalore from 2011 to 2013 before he joined CCMB as a senior scientist. And then currently his lab is extensively using genome edited stem cells and mouse embryos to study the cell fate decisions in the pluripotent state and self-organization of stem cells stem cells into the embryo-like structures. So with this, I welcome Dr. Chandrasekhar once again to this faculty development program. The audience will be really looking forward to your talk, and we wish you good luck on this session, so that we have many questions from the participants. Thank you so much. Over to you, Dr. Chandrasekhar.
Thank you. I'll first share my screen.
Yes, please, sir.
Is it visible?
Yes.
The presentation is visible?
Presentation is visible. You can keep it in the slideshow board.
Okay, that's fine.
The slideshow mode has not come in.
Full screen, sir.
Has it come on the slideshow mode?
No, not yet.
It shows slideshow mode in my system.
In your computer? Okay, okay. So
wait a minute, I would probably change it.
Oh, yes, now it is perfect.
I'm going for
no. Yeah. No. You have to get in once more. Yeah.
Now is it fine?
No.
Not yet is shared with the screen, sir.
Yeah, just share your screen.
Yes, sir. Go to the PPT. Yes.
Yes,
Is it
now
okay now?
perfect. Absolutely.
Okay.
Thank you. Thank you.
Thank you very much. Okay. On the outset, I thank Dr. Jaya and the organizers of this meeting for giving us an opportunity to talk some of our work. As you have mentioned, keeping in the mind the background of the audience, I'll try to keep it as simple as possible. And the audience can stop me anytime by raising their hand and ask questions, or they can keep their questions to the end, whichever way it is fine for me. So, as Dr. Jaya said, my lab works on understanding how the cell-fate choices made in the stem cells, particularly the early embryonic stem cells and the development of early embryonic stages in mammals. We particularly use mouse embryos as model systems, as well as nowadays we are also developing human stem cell based model systems of embryo to study the human development as well. Now, that being said, so the basic biology we do involves a lot of genome editing and changes in the gene to understand how the basic biology of pluripotent stem cells is achieved and how the cells undergo specific cell fate choices. Just briefly introduce you to the concept of these pluripotent stem cells. These mostly are derived from the blastocyst stage of the embryo, which can be cultured in the petri dish infinitely for as many passages you want. And also, when appropriate conditions are given, they can differentiate to any type of cells in the body. So they hold the potential to become any type of cell in the body. So they provide a great opportunity, one, to study development as well as for regenerative medicine. And one of the properties is called self-renewal, that is they can perpetuate and they can differentiate all sorts of cells in the body. Okay, so we use genome editing tools. Since this talk is mainly on the genome editing, so I'm trying to focus only on that. We use it extensively in the stem cell biology and in the mouse genetics to understand particularly how the gene functions affect specific biological processes, or to even understand the function of certain genes. So to just put it in a nutshell, why editing should be done in, or we do it in the embryonic stem cells. One is to understand the stem cell biology itself, that how the gene networks or genes regulate pluripotency. Then this is also utilized to have developmental biology studies to understand the function of genes and pathways during development, and to generate knockout mice and rat models, which work as very good disease model systems, and to study the human disorders in the mammalian model systems. And we also use it to develop reporter systems, which can create, where we can create assay systems for modeling diseases as well as for therapeutic applications using iPSCs. And we also use this for drug screening of iPSCs to or iPSCs derived cell types to create assay systems to screen the drugs. So I'll just talk to you about what are the possibilities of using the site-specific nucleases or the genome editing tools. Genome editing tools are basically the site-specific nucleases which can localize exactly a particular point in the genome, and most of the times it is used to create a double-strand break, but otherwise it can also be utilized for many other things. But what we have to appreciate here is it is a site-specific nuclease mostly, which can properly recognize, localize into a specific site in the genome. And the genome is really big. When we talk about human or mouse genome, it is around 3 GB genome. To localize to a very specific site of maybe, let's say, 20 bases or 25 base pairs, that means it has to have a very precision. So multiple site-specific nucleases have been generated. The first one to come was the zinc finger nuclease. Then came talons, and CRISPRs are the last ones to come, and they are quite popular. And the reason they are popular is they are very easy to use and very easy to design. And they are also free source that you can get the plasmids and build your own CRISPR system very easily. So the possibilities of using this site-specific nucleases is you can add or delete transcription. I'll just put a laser pointer. Okay, if you can see the laser pointer, you can add or delete transcription factor binding sites. That means you can manipulate the promoters. You can delete or add promoters, insert a constitutive promoter or inducible promoter, so you can bring about changes in the regulation of the gene. You can disrupt the gene function itself. That is what we call as knockouts. You can create a point mutation where you can make a specific amino acid change or a base change, which is similar to a disease counterpart or a diseased allele. You can fuse reporter genes if you want to know where this particular gene expresses in which tissues or which cell types. You can put a reporter gene like a fluorescent tag or a galactosidase, beta galactosidase site or a sequence, and you will be able to track the expression of the gene. And also something called a safe harbor site integration. These are mostly done in those regions where the genetic manipulation in those regions will not affect any gene function or do not give any phenotype. So such sequences are there in the genomes, and one can use those to put in whatever the external transgene one wants to put. Also to create conditional knockouts where you can flank or put something called as LOXP sites on either side of an exon and use it to delete. You can create or insert new exons, only remove one exon, or induce or put repeat elements of particular amino acids. All those things can be done. You can fuse a reporter gene in the end of the gene. You can fuse it to the protein that the gene produces, so that you can even track the protein that is produced by the gene. Also, you can delete or add microRNA control elements in the 3' UTR so that the regulation of the mRNA itself can be altered. So basically, using this sort of genome editing tools, you can do multiple things in a gene. You can manipulate multiple parts of the gene for different purposes. And also, not just one, you can actually manipulate multiple genes at a time using the genome editing tools. Now, coming to the CRISPRs, this is the one that we use specifically in the lab. You will be hearing about it a lot in the course of this meeting. So I'll just hint you about briefly what it is. So it's basically a combination of a nucleus and an associated RNA that is called, in this case, the single RNA, which is actually a combination of tracer RNA and a cRNA, where the green part, what you see here, is a variable region. That means you can change that. Whereas the orange part, what you are seeing, is the constant part of the single guide RNA. This part is recognized by the Cas9 and it forms a complex with the Cas9 protein. I am only talking about only one type of CRISPR system, that is Cas9 system, but there are multiples which others may cover. So this part, which forms a stem loop structure, is essential for its binding to the Cas9. Whereas this green part is something that provides specificity to the specific region in the genome. So this can be designed and altered at will, and by just, you can order oligonucleotides and clone them, and you can change this part at your will. So if you want to target a gene A, you can have a guide RNA with this green region that is specifically designed for the gene A. Or if you want to target gene B, then a specific guide RNA with this sequence that is specific to the gene B. Whereas, as I said, the orange part remains constant. So the way this operates is, this RNP complex, that is the Cas9 and the guide RNA, once they are inside the cell, they scan the the DNA and recognize something called as PAM sequence or protospacer adjacent motif. In case of Cas9, it is NGG. Different CRISPR systems will have different PAM sequences. So in case of Cas9, it is NGG, N followed by GG. N is any base followed by GG. This is what is recognized. Then it tries to unwind the DNA and tries to see the base pairing. If the base pairing complementarity is perfect, then it completely unwinds it, and then there is the nucleus gets activated and creates two cuts here. One is the HNH, and what you see below is RUVC. So this creates a double strand break. And this is what CRISPR does. What happens afterwards in the cell is left to the cell. It depends on the conditions that prevailing in the cell. So what CRISPR does is, using a guide RNA as an address, it localizes to the specific region in the genome and creates a double strand break. Okay. So once, let's say we are targeting a gene A using a guide RNA, once there is a double strand break, there are multiple possibilities in the cells. The most predominant possibility after a double strand break is something called as NHEJ, which you are definitely familiar with, non-homology enjoining repair. So in this case, entire set of NHEJ machinery is recruited to repair the double strand break DNAs wherever it is. It will try to rejoin them back, and this is an error-prone repair. So when this rejoining happens, there could be errors. That is, a few bases may be missing. Sometimes no base may be missing. It might be a perfect joining, but most of the times few bases will be missing or few bases will be added. By that, it creates a small change, or these are called as indels. And if your guide was targeting an exon of a gene, you would have created some insertions or deletions. And if this insertion or a deletion of few bases creates a frame shift in the sequence of your exon, what happens is, then there is frame shift, the frame shift happens, and this will create an RNA that will have a premature stop codon, which will undergo nonsense mediated decay. So just by changing even one base, deleting one base, or adding one base, you can create frame shift mutations, which will create an mRNA, which will have a premature stop codon, which will undergo nonsense mediated decay. Hence, although the mRNA is produced from this gene, the protein cannot be produced, and the gene is considered to be knockout. So it will not be able to produce a protein. So the simplest way to create a knockout is just create a cut in the exon, and if there is a frame shift created, then the gene gets knocked out. The other possibility, which is a rare possibility, in some cells it happens in higher frequency, in some in the lower frequency. Nonetheless, it is the rarest possibility, that is the homology dependent repair. In the homology dependent repair, you have to provide the donor template from outside. In case of NHEJ, when you want to knock out a gene, you don't need to provide any template. You just provide the Cas9 and guide RNA. Whereas in the homology dependent repair, along with Cas9 and guide RNA, you also provided repair template or a donor template. If you want to create a point mutation, you can have a donor template with the point mutation, which is indicated in red, or you want to insert a chunk of DNA or a gene sequence, which is shown here in the green. You can give a donor template, which will have homology arms on either side, the blue regions, which are complementary to either side of the cut in the genome. So this homology regions will trigger the homology dependent repair, and the homologous recombination happens, and the intervening sequence between these two homology regions will be incorporated into the genome. So in both these cases, this is how a precise insertion is created, where you may insert a few nucleotide changes, or you may insert a long gene or a coding region of some reporter gene or whatever. So this is the difference. So to create a knockout, it is pretty simple. Just to create a double strand break is sufficient in the coding region. Whereas when you want to insert something, you have to provide a donor template, or the we also call it as targeting vector in our mouse genetic terms. Or okay, so you have to provide a donor template or a targeting vector, which will be inserted into the locus. I hope this thing is clear. Now I'll just come to certain aspects of the work which we have done on the sidelines, just to improve the efficiencies in our case, to aid our studies. We don't work on CRISPR or CRISPR-Cas9 or CRISPR tools. However, we use them extensively for our studies of stem cell biology and early embryonic development. So one of the major hurdle we faced with respect to using CRISPRs was ES cells are very small, and they are very difficult to transfect cells. So poor transfection efficiency is one thing we realized is a major drawback in many cell lines where you can't get efficient editing. And also the time and the cost of genotyping, because if your efficiencies are poor, then you have to screen so many clones to find your precise changes. So we started fixing these things using different methods of nucleofections to and transfections to come up with a method which will give us a very good efficiency of transfection. We have utilized electroporations, lipofection, and nucleofection. The claimed success rate of nucleofection is 80%, but we did not see that when we use larger plasmids, this is the case. So we worked on different recipes of nucleofection buffers and conditions, and we came up with a method which can give us more than 70% efficiency, even using very large plasmids like 9 kb, 10 kb plasmids. The previously claimed 80% is only with a 3 kb plasmid. It doesn't work for large size plasmids. So first we sorted this out, and we have come up with a system where we can do a very high efficiency transfection. Then using this transfection, these are our own buffers which we have generated. Then to increase the efficiency of targeting and create an easy method for creating knockouts, what we designed is something called a twin guide strategy, where we always design 2 guide RNAs for a gene to be knocked out. So I said that you can design a single guide RNA into the exonic region, and if you make a double strand break, there can be a frame shift that creates a knockout. That is true. But if there is 1 base insertion or 2 base insertion, it is very difficult to detect it. You will have to amplify in sequence. You can't do that for every clone. So what we decided is we will use 2 guides, and then we decided design primers on either side of the guides, so that when we amplify the wild type, which is shown here, let's say we get 560 bases. If 2 guides are working at a time, they would delete the intervening DNA region in between, so that the PCR will show you a shorter band, and you know that the deletion has happened. So using this, we have generated a lot of knockouts. So what we have done is we have created this plasmid called twin guide plasmid, where you have 2 U6 promoters, and this is the U6 promoter driving the blue region is the guide RNA part, and another U6 promoter driving another guide RNA part. So in between this promoter and this one, we can clone our guides. So 2 guides it can carry for the same gene. So using this plasmid, what we also understood later is we could use it for multiplexing, not just 2 guides. We can actually clone multiple guides. In this case, we can clone up to 4 or 6 guides, and you can target multiple genes at a time. So 1 plasmid can be utilized to target multiple genes. So you can multiplex CRISPRs. And also we use different fluorescence receptors like IRFP, GFP, and BFP. So we have multiple constructs, so that when our cell line already has one of the fluorescent reporters, we can use something else. So we have a huge toolbox of these plasmids created, and also the system that is established for creating these plasmids carrying multiple guides. So we use mostly Gibson Assembly for this purpose. Some of you may be aware of it, but I'll just try to, for the benefit of the students, I'll just try to explain. So Gibson Assembly basically is one of the cloning systems which works little differently. So the composition of this, it has a T5 exonuclease, then a proofreading polymerase, and tag ligase. These 3 are in the same reaction, but tag ligase you may or may not use. It doesn't make a difference as long as you are using E. coli, which contains its own ligases. Then the reaction is an isothermal reaction which happens at 50 degrees centigrade for 15 minutes to 1 hour. I'll tell you what happens meanwhile when you set up this reaction. So this is how a typical Gibson assembly reaction works, where what we would have done is we would have created PCR products which will have overlaps regions. So for example, I want to join this pink fragment with the green fragment, then I would have by PCR introduced a region which has complementary region. For example, the 3' end of this pink region will be complementary to the 5' end of the next fragment, that is the green fragment. Okay. So when I have put equimolar concentrations of these in the tube, the T5 exonucleases, it is a 5' to 3' exonuclease, so it will start eating one of the strands and creates an overhang. So you can see this, and they find the complementarity and they hybridize. After this, the fusion polymerase or any other polymerase, which is a proofreading polymerase, will come and add back the nucleotides, and it will rebuild the DNA. And whatever the nick is left behind, that is repaired by Taq ligase, or if you transform the bacteria, it will be repaired in the bacteria itself. So this is typically how a Gibson assembly kit looks from NEB, where you can have your vector and you want to clone these two fragments in tandem. So you can use this in a single reaction, you can clone both the fragments. So using this, you can clone up to 6 fragments at a time. In our hand, 4 works pretty well, but there are labs where people use even 6. There are labs where people have done even 6 fragments at a time. So what this allows is, so in this, what I'm trying to say is the fragment A and B, what you're seeing are our guide cassettes. So we can have multiple cassettes of this kind, and we are able to clone multiple guides in one shot. Okay, that's Gibson assembly. So these constructs, whatever we build, then we have to transfect them. And once we transfect, as I said, the transfection efficiency is not great in ES cells, or in whichever cells the transfection efficiency is not good, one can go for utilizing FACS to sort the cells which are transfected. For example, here on the left, what you see are untransfected control, and in the right, you see is the transfected control, where you can take the transfected cells, sort them separately, and plate them. So you know now that all the cells which you have taken have the plasmid, and mostly the plasmid is transfected in the form of a supercoiled plasmid, because you don't want it to integrate, you want it only to express the Cas9 and guide RNA. Alternately, if FACS is not available, we have puromycin resistance cassette, which works very fast, and a selection of around 36 to 48 hours is sufficient to kill all non-transfected cells, and whatever leftover cells are there, they can be plated to form single cell colonies. So in ES cells, each cell forms a beautiful colony as shown here. This may not be the case in many other cells which are mobile, but there are many cell lines where they may form colonies. So it's pretty easy for us to pick up a colony. We can physically pick this colony and passage it into a single well. So likewise, each colony can be picked up and passaged into a single well. Whereas in case of those cell lines where people can't have this sort of colonies, what people can do is trypsinize the cells, go for a limited dilution, and plate them into a 96 well plate in such a dilution that you get 1 cell per well, and later on that can establish into a colony. And once the colonies are established and clones are established in the 96 well plate, one can isolate the DNA and look for the specific genomic change that they have been intending to look for. So what we do after this is isolate the DNA. So we replica plate these clones. So they are always in 2 copies. 1 copy we culture and maintain it. The other copy is utilized to make the DNA. Then the DNA is utilized for, genomic DNA is utilized for doing the genotyping PCR. Here, I want to show you the example here. This is a typical, let's say, wild type band that comes at a specific size. So what you see here are the wild type bands. If there is a knockout, as I said, there will be a deletion. A small part of the DNA will be gone. So there is a deletion here. So there are 2 bands. That means these are all diploid cells. That means in one allele there is no deletion. In the other allele there is a deletion. Okay. For example, if you see this last clone here, which is shown in the red star, here what is happening is you don't have the upper band at all. You only have the lower bands, which shows that both the alleles have been edited. That means it's a homozygous knockout. So the advantage of this system is you can not only pick up the knockouts easily, you can also pick up which is heterozygous and which is homozygous. This is another example here down there. So that's how we create the knockouts in the ESLs. So this is an example to show you how exactly it looks like. So if, let's say, these are the two primers we use for PCR amplification, this is the size of the amplicon that comes in. And this is my guide. If my guide, so in CRISPRs, always the cut happens, Cas9, the cut happens 3 bases before the PAM. This is the PAM sequence here, ACC or TGG. TGG is the PAM. It is reading this side. And 3 bases before, here is the cut. And here, the PAM is AGG in this frame, and the cut happens here. So this is the part that will be deleted from the chromosome. So if you use these two primers, the DNG 36 and 37, you get a specific size. If the deletion has happened, you will get a smaller size. So by that, you know that the deletion has happened. This is exactly how we genotype it. Okay, using this twin guide strategy, we knocked out multiple genes. And what we found is, depending on the gene and the strength of or the specificity of the guide RNAs, we get a deletion efficiency or a knockout efficiency of 7 to 14%, which is decently good for us, because otherwise the knockout efficiency in ESLs varies between somewhere between 0.5 to maybe sometimes 3 to 4%, but not more than that. But here we can get really good efficiencies. In certain cases, we have even got 40–50%, but those are outliers. But this is what general efficiencies are. Okay. Now that is how to create a knockout. Now I'll talk about how to create knock-ins. So in case of knockout, you are deleting a gene function either by deleting few bases or the whole gene or deleting a specific part of the gene. In case of knock-ins, you are going to put in an external DNA into a specific site in a specific manner. Okay, it is not transgene. In a transgene, you just put the DNA, it will go and sit randomly somewhere in the genome. Whereas in a knock-in, you will precisely go into the locus and you will make a change which you precisely wanted. No randomness is allowed in this case. So how a reporter knock-in is made using CRISPRs. I'll give you one of the examples that we have used in our lab. We wanted to tag GFP to one of our genes called NANOG, and we wanted to track how the NANOG protein behaves during certain treatments or differentiation. So what we did, we designed a guide RNA which will cut in the last exon of NANOG, and it will encompass the stop codon of NANOG. The reason we did this is exactly after the last amino acid of NANOG, we wanted the GFP to be fused to it so that we can track the NANOG protein live in the cells. So we used a guide RNA that will cut overlapping the stop codon, which is TGA here. Then along with that CRISPR plasmid, we put a targeting vector. I said that if you want to make a knock-in or a precise change, you have to give a donor template. So in this case, we provided a targeting vector which contained GFP, iris, and neomycin. So GFP is what we wanted to fuse it. Iris is interchromosomal entry site, which creates a separate translation from the same mRNA. So you can have neomycin expressed as a separate protein, although it's the same mRNA. Just behind the GFP, we have the last exon and the fourth exon of NANOG coming as a left homology arm. It depends on how long the arms are. Okay, I'll just tell you on that. And on the right side, you have another homology arm, which contains the 3' UTR of the gene and the sequences beyond the NANOG gene. Now, what is the size of the homology arm that is utilized here? So in case of bacteria also, the homologous recombination happens. In case of bacteria, you just need 50 bases of homology arm, and it creates a beautiful homologous recombination, and it's very easy to knock out the bacterial genes, particularly in E. coli. In case of mouse, previously people used to do these knockouts without CRISPRs, where we have to build at least 5 to 6 kb of homology regions. Without that, the knockout efficiencies were undetectable. Whereas using CRISPRs, the CRISPRs will create a cut here. So you can actually succeed. We have used as small as 125 bases of homology arms and it works. So your targeting vector can be really small, and as small as 125 bases of homology region on either side is sufficient to create homologous recombination if you are using CRISPRs. The increase in the efficiency is just because there is a cut that triggers homologous recombination in this site. And if this plasmid or DNA is found around, it will be able to stitch it inside. Okay, so we co-transfect the CRISPR plasmid as well as the targeting vector at a time into the cells. So whichever cells carry both of them, in those, the homologous recombination can happen. So we could create multiple cell lines like using this system. Okay, this is just an example that once, so this is a FACS profile of what you see in black is the wild type cells, which don't have any GFP expression or IRFP expression. What you see on this side, the right, are the ones that start expressing. So they express at different levels depending on different treatment. So one could easily create fluorescent tagged proteins. So like this, you can create any tag, not just GFP or IRFP. You can create a flag tag or any other tag of any other protein. So the method involves the same process and same principle. Okay. So how the genotyping of this? See, one is introducing this change and how to detect the change. The most important thing in any genome editing is, okay, one is you have an efficient system to make the change. You also should have an equally efficient system to detect the change. If not, you will spend a lot of time searching for which is right and which is the wrong event. So the way we do it is we design a primer from the cassette which we are inserting. For example, this is the cassette from GFP, a primer that is reverse primer that can go this side, which is shown here. And another primer comes outside the homology arm. So this is the homology arm. Any primer outside this region, which is shown here from exon 3. So the amplification will come only when your cassette is sitting in the locus, because the reverse primer will bind here. But if the cassette is not sitting in the locus, the forward primer cannot amplify it. So this amplification will come only when your cassette is inserted into the specific locus. That is what you will see here. So we can use a simple PCR to say that if it's knocked in or not. But this doesn't distinguish that if both the alleles are knocked in or not, then you will have to do one more round of PCRs to know if both the alleles are knocked in or not. That is, you will see that the other allele is not wild type, but it has also converted. That's when you can say. But otherwise, this is a simple PCR strategy to genotype and pick up this specific insertions or the knock-ins which we do. Okay. So using this, we have created multiple types of lines which can give GFP, RFP. One allele is GFP, one allele is RFP. So we can study how each allele behaves. Okay. So what lessons we learned during this study or making these lines is, traditionally it is known that it is the linear DNA that triggers linear double-strand break DNA, or the DNA with free ends of DNA. That is the one that triggers the DNA repair system, and that's how homologous recombination happens. However, in our study, what we found is, if you use a supercoiled plasmid, the efficiencies are much higher. Okay, that was surprising in the beginning. We couldn't explain it because you would expect that you need a free or a broken DNA to trigger the DNA repair system. But later on, what we understood is, if we co-transfect a supercoiled plasmid of the Cas9 and a linear plasmid of the donor, what was happening is, even before Cas9 is activated and makes a cut in the specific site, the linear DNA is integrated elsewhere. Whereas when both were kept as supercoiled plasmids, the targeting vector or the donor DNA persists in the cell till there is a double strand break made in the specific site, and then the DNA repair system is triggered in that specific site, and that will pull in the supercoiled plasmid of targeting vector and insert it into the locus. So this is what we found, that you can increase the efficiency of these gene targetings and knock-ins using supercoiled plasmid. So the efficiency is more than 8 to 9%. We have done a lot of experiments in a lot of loci, and this is what it turns out to be, which is a very good efficiency compared to less than 1% which we normally get. So this is another example I'm showing where we have used it to in a different cell type, and we were able to knock in and show that we could knock in a GFP into another site, a different cell type called SPGC1 cell lines. Okay, so and where we got a 14% efficiency. And so the take-home message from these is that using twin guide strategy, we can mostly make precise deletions. So also another thing which we found in this study was when we use a single guide, we cannot predict the outcome. When we were using two guides, what we realized is mostly the DNA was joined exactly where the cuts had happened. So in 70% of the cases, we could predict exactly how the deletion will happen. So that gives us a little better handle that we can make precise deletions. If you want to only delete few amino acids, we can do that using this. Whereas that is not possible using a single guide. So we don't exactly understand why this precision comes in when we use two guides, but that is something we have observed. And also, it is very easy to genotype. And we also show that using a supercoiled donor vector actually enhances the efficiency. Again, as the convention that we have, that it is the linear DNA that enhances homologous recombination, but using CRISPRs, we realize that it is supercoiled one that works better. So we have also used something called negative selection markers, which I didn't discuss in detail, but we haven't, we don't see a major change. Sometimes we find twofold increase in specific sites. Okay, otherwise you can do away with even negative selection cases. Okay, now taking you a little ahead, this is till now I talked about how we make a deletion or a knockout, or how we make a precision change to knock in a specific sequence or a gene of our interest into a specific locus in the genome. Now, what I'm going to talk is few of the CRISPR modification systems which we use to carry out our studies. So one of the early systems that came in was the CRISPR interference system. So if you go back and recall, I said that the Cas9 creates a double strand break. So that is what is the basis for creating a knockout. But later on, people came up with Cas9 protein, which is nuclease deficient. So the nuclease deficient, basically, if you just recall back, I said HNH and RUVC. These are the two catalytic domains of Cas9. Each one of cuts in one strand. So if you make a specific point mutations in these two places, RUVC and HNH, you can create a CRISPR-Cas9, which cannot cut the DNA, but it can still bind to the guide RNA, and it can go and specifically locate to the specific sequence in the genome and unwind the DNA, but it cannot make the cut. So what is shown here is using this system. These are called as dead Cas9 or nuclease dead Cas9 or nuclease deficient Cas9. So this in no way affects the ability of the Cas9 to recognize its sequence and open up the DNA. That is not affected. The only thing that is affected is it can't create a double strand break. People have also created only one mutation and created nicases. That means it can create only cut on one strand. So those systems also exist. But for the CRISPR interference, what is used is the D-Cas9 or nuclease dead Cas9. So if you use a guide RNA that targets just below the TSS or the transcription start site, what will happen is the Cas9 will come, open up the DNA and does not allow the polymerase II to progress. So it can block elongation of polymerization or transcription. So by this, you can actually knock down or tone down the expression of the gene, a target gene. So this is called as knockdown. So the difference between this and the knockout is, in knockout you are making a physical change in the target DNA. Here you are not making any physical change in the target DNA. What you are doing is you are using the guide and the Cas9 to open up the DNA just below the transcription start site, so that the polymerase will find a steric hindrance to move and it will not be able to progress. So this will affect the transcription and decrease the transcription by 90 to 95%. So this is a knockdown. Alternately, one can also have guide RNAs designed for the transcription initiation site itself, so that the polymerase is not able to load onto the gene, onto the promoter, and it prevents the initiation itself. You can have multiple guide RNAs, one blocking initiation, one blocking elongation, and achieve a very high level of CRISPR interference or knockdown of the target genes. Now, this phenomenon also can be multiplexed. You can have multiple guides for one gene and block it efficiently. You can have multiple guides targeting multiple genes. Let's say one whole pathway you want to shut down, you can have some 7-8 guides expressed in the cell, and all those genes can be shut down at a time. So it is not a complete knockout, it is a knockdown. That is something you will have to distinguish between. And if your Cas9 can be removed from the cells, it can be reversed easily, whereas a knockout cannot be reversed. You will have to do another editing process to revert it back. So that's the advantage of CRISPR interference, where people want to target multiple genes or genes involved in specific pathways, and knockdowns are one of the easiest ways to start it. Alternately, there is also a system of CRISPR activation. So in case of CRISPR, so just a minute, just go back to the slide. So if you want much more effective blocking of the CRISPR interference, what people have done is people have also tagged this Cas9 with a transcription repressor domain like CRAB domain that will bring in the repressor transcription repressors and repress the gene. So that is one step above. So you can not only block the polymerase, you can also bring in repressor complexes and completely block the transcription. So on the same lines, CRISPR activation was generated. In case of CRISPR activation, you have a dead Cas9 that is tagged or fused to the VP16 domain, which is a transcription activation domain. So this VP16 is a domain to which the transcription activators can bind, and it can bring in the polymerase 2 and activate a gene. Okay, so you can use the dead Cas9, same dead Cas9 to activate a gene. You can use the same dead Cas9 to repress a gene, depending on where your guides are targeted and depending on what is fused to this Cas9. If CRAB is fused, it will repress. If VP16 is fused, it can activate. However, here the guides will be targeted little differently. The guides will be targeted to the promoter region only. They can't be after the TSS because that will block the Pol2. So mostly it is targeted between upstream up to 300 or 400 amino acids, and the guide will guide the Cas9 to sit here, and the VP16 domain will bring in the transcription activator complexes, and Pol2 will be loaded, and that will lead to active transcription of the gene. So even those genes which are silent, which are not expressing, one can use this system to activate the genes, or some gene that is expressed at low quantities, you want to express it in higher quantities or increase its expression, one can use that CRISPR activation system. So you have both the systems which can interfere. So what we did is we use these both systems and try to build a single system where what we wanted to study is we wanted to simultaneously activate and inactivate a specific set of genes in neighboring cells. What I mean to say is, in a culture, let's say I create a cell type, then if some 10 cells are there, I want to activate gene A in, let's say, some 5-6 cells and inactivate the same gene A in another 5-6 cells. So it will be happening in different cells, but in the same culture I want to do. So for that, what we created is we combined the CRISPR-on and CRISPR-in system. The CRISPR-on was tagged with RFP, and the CRISPR-in was tagged with a GFP, what you see here. And we put them in opposite orientations. Basically, the idea is at a time, either the cell can have CRISPR-on or CRISPR-in when it is flipped around, or it can be turned around. I'll explain you how it works, but this is using Cree-Loxp system. Okay, so let us say once we have created this cell, this is what we have created. Now what we have is the CRISPR-on system in here cells. To begin with, the cells do not express either CRISPR-in or CRISPR-on. Okay, and when we add tamoxifen, which is a component, which is an analog of estrogen, what it does is it will flip the CRISPR-on or CRISPR-in, which is driven by a tetracycline promoter. Okay, there is a tetracycline promoter which currently faces the CRISPR-on, but when you put tamoxifen, it can go exactly opposite. The CRISPR-in can come here and CRISPR-on will come in the other orientation. So that is what we can create. Once we put tamoxifen, some cells will have CRISPR-on facing the promoter, some cells will have CRISPR-in facing the promoter, but they are not on because they are under the doxycycline controllable or tetracycline controllable promoter. When we add tetracycline or doxycycline into the cells which had CRISPR-on, now they become red because now the promoter is on and they start producing CRISPR-on system, that is Cas9 with VP16, and the CRISPR-in cells which are green in color, now they will produce Cas9 with crab domain. So if I have a guide RNA targeting gene A, in CRISPR-on, the gene A will be activated. In CRISPR-in, the gene A will be repressed. So at a time, we can create cells which have a knockout in some cells and a knockdown in some cells, and induction of the gene in other cells. We can also select them separately by using puromycin and G408 because the CRISPR-in can be selected with neomycin and puromycin can be used to select the CRISPR-on system. So using this, we targeted a gene called OCT4, which is actually a pluripotency factor. What we see is when we are able to knock down OCT4 using CRISPR interference, the cells will differentiate to trophoectoderm, which is one of the layers of the embryo, which forms the placenta. Whereas when we induce OCT4 using CRISPR-in, what happens? We get cells which are from primitive endoderm, which will form the yolk sac of the embryo. So basically, the fate of the cells we can change by changing the amount of OCT4, and this we can do using the same cell system which contains CRISPR-on and in the same cells. Okay, this is one of the examples, but we have multiple such examples where we study the cell fate by altering the expression of certain pluripotency factors and see where they will lead the cells to. Okay, now that's about the cells. Now we will go into the genome-edited mouse models. So I'll take you briefly through the process of creating genome-edited mouse. So traditionally, the knockouts were made. They are being made from at least past more than 3 decades. The traditional route was embryonic stem cells are cultured in the culture, and you make the mutation first in the embryonic stem cells using homologous recombination, whatever I talked about there. Except that previously there were no CRISPRs or genome editing tools. We used to use homologous recombination. That means the constructs were really big, the targeting vectors. So you make the change first in the ES cells. Then once you confirm that the mutation has happened in the ES cells, take these cells, inject them into the early blastocyst. So these cells, since they were derived from the inner cell mass of the blastocyst, they retain the ability to form all the cells in the body. They will incorporate inside the this blastocyst, And once these blastocysts are there, they are transferred into a pseudopregnant mouse. That is a mouse which is prepared by hormones to carry the pregnancy. Once we put that, in many cases, these cells are able to successfully integrate into the inner cell mass of the blastocyst, and they give something called as chimeric mouse. It's a chimeric mouse is basically a mouse that comes out of contribution from some cells from the embryonic stem cells which we have put and the stem cells that are coming from the blastocyst host. So it is called chimera, and it can be easily detected because the coat colors are different. So the ESLs what we have are coming from a strain that will have agouti coat color, that is, let's say, brownish. The embryos which we are using are from black 6 mouse, which is black. So the chimera will have both brown skin as well as the black skin. So it will have both brown and black hairs. That is how we know that it is a chimera. So that means the contribution of the mutated stem cells has happened into the chimeric mouse. Now what we do is this chimeric mouse is again bred to see if the mutated ESLs not only contributed to the skin, but they also contributed to the gonads or not, to the germ cells, because the germ cells will be passed to the next generation. If in the mating we start seeing the brown mouse, which suggests that this brown color is coming from the ESL component that we had injected. That means those will carry the mutation now. Now, once this is there, this is a heterozygous animal, then these can be mated to obtain homozygous animals. This is how a typical knockout is created. And the time span is, it can run into a couple of months if you are lucky. If you are not lucky, it can even run to a year or more. Okay, first one has to create the ESLs and cross-check them, verify that they have the mutations. Then you make chimeras. Some chimeras will carry the mutation in germline, some will not. So you'll have to wait for them, or you have to create multiple numbers of them. And then only you will get a germline. And from that, you will have to generate the homologous. So it takes 6 to 8 months or to 1 year. Now, what has happened is, after the CRISPRs came, this process has become very simple and very easy. So you can now create mouse, knockout mouse using the same system, except that in ESLs, you can use that and create more number of ESLs at a faster rate. That's one thing. Also, alternately, you can directly create knockouts by injecting CRISPRs into the embryos of the mouse itself. So what one can do is, can inject the Cas9 complex along with the guide RNA into the, this is to create a knockout, into the pronuclei of the zygote. So once both the nuclei are there, you can inject it into one of them. Now what can happen is there can be multiple outcomes. One, as I said, there is no activity of CRISPR in the first stage, so you will get nothing in the first 1-cell stage. But the CRISPR might act at a 2-cell stage, and in one cell there can be a complete knockout, and in another it might not be. This will create something called as mosaic. That means in the body of the animal that has come out, some cells will be knockout, some cells will not be knockout because they are coming from 2 different types of cells. This happens just because the CRISPR acted little late. Otherwise, the CRISPR can in most cases act very early on, and what you will see is a knockout, and many times it can be homozygous knockout. Both the alleles have been knocked out here. For example, let us say if we have used a gene which controls the coat color, and if you knock it out, it will become white. If it is mosaic, you will get black and white patches. Or it might happen that only one allele gets knocked out and the other is not. So if it is a dominant phenotype, if it is a dominant allele, then what you will see is it still exists as a black one because the other allele is taking care of the function. So in this case, it is a heterozygous which you will have to breed and then get the homozygous out of it. Whereas you can directly obtain homozygous in many cases. So this system, one, it works fast. It is less expensive because you don't use ESLs. ESLs cost a lot of money to grow. There is, and it also takes a lot of time when you go the ESL route. You have to create a chimera and then create a germline, then you make the homozygous. Whereas here, there is a good chance that you can get the homozygous in the first shot itself. So this has changed the whole dynamics of creating knockouts in mouse. Not just in mouse. This is an example I am giving from mouse, but this is the same system that is used in rat. This is used in now multiple model organisms. So that's another big difference that CRISPRs have created. That is, the knockouts were only possible in mouse before. In rat, it was rare. 1 or 2 papers were there and they were extremely difficult to create. And from other higher organisms or model organisms like rabbit and all, it is unimaginable. But once the CRISPRs have come, now knockouts have been created from every sort of animal, including the large animals like pig, cattle, everywhere. So this has made the things pretty easy and pretty straightforward. And particularly in those model organisms, or even in the organisms that are utilized for commercial purpose, what is happening is this has reduced the time of generation of these knockouts. So particularly in large animals, if you think for this sort of a mutation to be created, it will take a decade or so, just because the breeding cycle takes such a long time. But you can create these things much quickly using CRISPRs. So that, I think, is one of the biggest advantage that it has created, that almost the editing can be done in any sort of organisms. Okay, so this is just another example I'm giving from our lab where we tried to knock out a gene called ATD, and this is what you see that these are all the heterozygous knockouts which we found. So the deletions are not always precise. Sometimes they are different, but we do get 3 knockouts, and then they can be they give the germline transfer also. So what we do is, many people use the Cas9 and the guide RNA complex to inject into the cytoplasm. What we do is we use the same twin guide plasmid, which actually cuts cost for us, and it's pretty easy and simple for us to do. We inject it into the cytoplasm. Then the expression of the guide and the Cas9 takes care, which we can track with mCherry, and then the editing happens. So using this, we have created a couple of knockouts here. This is one of them. ATD is one of the knockouts. And then its germline transfer is pretty straightforward. So if you have this, even if you have mosaics, you can detect them easily. So once you have the animals created by genome editing, either you can use spermatogonia or the blastocyst stage, and you create the first founders, and the founders will be mated. If you have 2 founders, it is good. They will be mated to get a pure mutant animal. So this is the germline transfer to get a heterozygous knockout being passed from the ATD founders. So this is how we create the knockout mouse, and this is the same system that is utilized in any other organisms. So as I said, be it zebrafish, be it rat, be it rabbit, be it pig, in all the systems, exactly similar things happen. And as I said, CRISPRs only create the double strand break. They don't do anything after that. That is, their job will end there, and later on, the changes in the cell are dependent on the cells. Now, coming to the other type of modifications from CRISPRs, which are much more interesting and have been gaining more importance in the recent times, is the base editors or double strand break free genome editing. There are limitations to this, but these are fantastic tools. These are based on, again, dead Cas9. Here, what can be done is, one can alter a single base, maybe 2 or 3 bases, without creating a double strand break. If you recall what I was talking, to create a change in the DNA, you have to make a double strand break, then give a template or a donor template or a donor targeting vector to make a specific change. So this also had certain issues. Now, one was if the cut happens in a wrong place. So these are called off targets. So if a cut happens in the wrong place, then the DNA may go and integrate in a wrong place. So this is okay for the regular biology, but when you think of doing the gene therapy or therapeutics, that is not a desirable thing. So people were looking for systems which can operate much better without creating double-strand breaks. And also double-strand breaks sometimes can create chromosomal arrangement, rearrange the chromosomes or swap or create translocations. So people were looking for systems which can create change in the DNA without using any double-strand break. So then this came the base editors. So in base editors, what you will see is there is a D-Cas9, that is the nucleus dead Cas9. That is fused to, in this case, cytidine deaminase, where when you want to change a C to T, okay, that change you want to make, you use a cytidine deaminase. Just like what I said before in CRISPR interference and CRISPR-on, that the dead Cas9 doesn't lose its ability to bind to the DNA and unwind it. Okay, it retains all those capabilities. Now, what is tagged to it will decide what will happen. There, if you tag VP16, it will activate the gene. Here, if you use cytidine deaminase, and what you see here is this red part is cytidine deaminase, and this is a small linker that is attaching these two. Depending on the length of the linker, you can target a specific sequence here. So when this complex of D-Cas9 cytidine deaminase with the guide RNA is transfected into the cell, it will go and identify the specific region, unwind the DNA. Once the DNA is unwound, the cytidine deaminase will bind to this sixth base from this 5' region or this 3' region on the other strand, and it will deaminate it. So whether it does the fifth or the fourth base, that can be decided by the length of this linker that is. So people have created multiple base editors with different length of linkers. So you can specifically target one base. So it deaminates this, and once the deamination happens, this is an enzyme. Okay, once the deamination happens, now this is pursued as uracil. Okay, either the DNA repair system will try to repair it, or in the next subsequent round of replication, what happens? This is pursued as uracil or equivalent to T, so the opposite strand will be changed into A. Okay, by this, what was started as CG can be converted to TA. So you can get a C to T change very easily using cytidine deaminase. Okay, so then they also came up with another base editor that using adenine deaminase. So here it will have the same Cas9 with the adenine deaminase, this one domain. What it does is it will deaminate the adenine on the opposite strand, and this is converted to inositol, which will be pursued as G, and the opposite strand will be changed. Okay, sorry, it will be pursued as C, and the opposite strand will be changed to G. So you can get A to G transition. Okay, that's how you can make a single base pair change without any cut in the DNA. The advantage of it is, even by chance, if it goes and corrects in a wrong region, it will be only one base pair change. Okay, it won't create a double strand break. So it doesn't give opportunity for any chromosomal translocations or anything. So that makes it much safer. And people have now evolved Cas9s with very high specificity and very low off-target effects. So they can make precise changes in few bases. And this will permit repair of specific mutations, particularly in the disease cells. One of the best places one can think of is in like cases like sickle cell anemia, beta thalassemia, where you have a single gene disorder which is created because of a single base pair change. This sort of tools will be very handy to reverse those changes so that the mutation will be corrected in situ in the cells. Probably one can take out the bone marrow cells, create these changes, put them back, or one can have a delivery system which will be able to change the cells in the bone marrow itself. The sequence, these alleles can be reverted back to the wild type so that this disease can be taken care. So the base editors will have a very great value in the coming days, and they are already adopted in multiple places for gene therapy. And a lot of, I think, the trials and developments are going on. We use this only to study our basic biology because we have certain sites which are hotspots for certain transcription factors to bind. We alter them using these base editors so that we can alter the binding sites of the transcription factors and see what happens to the gene regulations. Okay. So not just one base, using multiple guides targeting multiple sites, one can repair or edit multiple bases in the same gene or in the different genes or different chromosomes using the base editors. Okay, so with this, I'll bring to end my talk. And this is my team, mostly PhD students, and this is our technician and some of the dissertation students here. So with this, I'll be happy to take questions from you guys.
Thank you, Dr. Chandrasekhar. That was really wonderful because it really has given a kind of hands-on. Though it was absolutely virtual, but I felt a bit of a classroom session, which was really needed because our participants would be looking forward to this type of technical sessions with real knowledge on how to design these sort of sequences, because that part is very important. So I think this session was really has made a good mark on that aspect. Before I go into the questions, I think there would be some questions from the audience's end. So can we take some questions? Prem?
Yeah, please.
Okay. Yes, Prem, please keep some, just check, and Shohini, please check with the questions.
And we have 2 questions in the question answer section.
There was some, I think, students, there were participants who raised hands even. So I will take the questions first, and then we will move on to those if they want to ask directly. Okay. So here, Dr. Chandrasekhar, there is one question. This is from Dr. Minakshi. She says, could you please explain how the CRISPR-mediated knock-in differs from conventional method of homologous recombination?
Okay, thanks for that question. So the only difference is that in case of conventional homologous recombination, you need a free DNA or a broken DNA to initiate the DNA repair. So that is provided by your targeting vector, but the repair can happen only when your DNA is unwound. So mostly the conventional homologous recombination happens at the S phase predominantly. Okay. Whereas in case of CRISPRs, what happens? The CRISPRs create a double strand break in the specific site where you want to create it. So at that specific spot, the DNA repair system is triggered. So when that happens, if the targeting vector is hanging around in the cell, that will be incorporated in. So this creates a higher efficiency just because the double strand break is happening at the site where you want the homologous recombination to happen. Whereas in case of conventional knockouts, there is no double strand break created at the site where you want. So it's a chance factor that your targeting vector is hanging around at the S phase near this gene. So that is where it will happen. Okay, that's where the difference in the efficiency also comes up.
Okay.
Otherwise, the mechanism downstream of it is exactly same. No difference.
Exactly. So Dr. Chittabrata asks that how long the Cas protein can block the transcription process?
Okay, that's a good question. Actually, that is dependent on the concentration of your Cas9 and the guide RNA in the cell. So if you have a very high robust expression of guide RNA and Cas9, so basically these are dynamic processes. They are not static processes. That means if I say the the CRISPR system or the CRISPR interference happens by binding of the Cas9 and guide and unwinding the DNA and blocking polymerase. If the concentration of Cas9 is low, it binds to the DNA, falls off, binds to the DNA, falls off. If its concentration is very high, its residence on the time on the DNA is more. So depending on the concentration of your guide, it can, or the CRISPR, it can block it more. If the expression is low, it will block it less. It is just because its residence time on the DNA is different because of its low concentration. So if you have a very good expression constitutively, then it can block effectively for a long time.
So how can that constitutive scenario be maintained? Means, is there any way to keep it stably expressed in high levels?
Yeah. So one has to put the Cas9 as a transgene under a very strong promoter in the cell, make a stable cell line, which constantly expresses the Cas9, as well as have 2-3 guides targeted to the promoter as well as the TSS or downstream of TSS. If you have 2-3 guides, then the interference is very effective.
Okay. So these promoters which we conventionally use in our cell lines to create those same plasmids we can use?
Yes. But you will know which promoter works well in your cell, so you use the most strong promoters.
Okay, definitely. But there's a question from Anuj. He asks that ethical issues associated with the mammalian. Are there any ethical issues when you are using the mammalian research system with respect to your technique? Are there any ethical issues?
Okay, so with respect to the mouse, there are no ethical issues. We these days use human cells to create human blastocyst-like structures called blastoids. So those things will have ethical issues, which anyway we get through the IEC, and we only have a limited window where we can work. Otherwise, mammalian systems, there is no serious ethical issues because we are using them for the same purpose otherwise which we would have used using the conventional methods. So I don't see any ethical issues in using them in the model organisms.
Okay. So Dr. Deepak asks that can you name the promoters? I think you have already answered the some of the promoters. There should be all viral promoters, right? The strong
so it
promoters.
Depends on the systems you use. For example, viral promoters don't work in our system. So in the early embryos and the embryonic stem cells, viral promoters are actually heterochromatinized. So we don't.
So there's depression. There'll be a depression.
So we use something like EF1 alpha and PGK, whereas I think CMV and all will work well in most of the other cell lines.
Okay, so that explains. So I have some few questions, Dr. Chandrasekhar. So if you permit, I can ask you.
Yeah, please.
Yeah, sure. So these are because your session was so technical and it was really I found it very interesting because when we are teaching students in our classes, we really need to make them understand. Now, I found that when you were explaining that Gibson Assembly, that part, so you mentioned the TGA, that the stop codon. I just want to double check with you that you must have bypassed or you must have removed the stop codon so that your GFP can be fused, right? Am I correct?
You're right. Yes.
Okay.
So in the target, we do remove that. Yes.
Yes. So this is for the understanding of the participants that the TGA stop codon had to be removed. Otherwise, there wouldn't be an expression of the promoter downstream. The reporter, the reporter downstream. Right.
And it is in frame with the protein.
In frame with the protein. But now this is something absolutely different. What is this nanoc protein and why did you choose this? Means you must have some ideas because we are just wanting to know. It is just out of curiosity. What made you choose nanoc?
Oh, okay. So that comes from my basic biology question. So nanoc is one of the 3 core pluripotency genes. OCT4, SOX2, and nanoc. Nanoc is called as also it's basically an Irish term used for ever youth. That means this gives the embryonic stem cells its capability to self-renew. So our lab actually studies this gene, and particularly it has got some very interesting expression. It has one of the very interesting gene regulatory systems. So this gene shifts from expressing from one allele to another allele. Depending on the dosage, it will go to 2 alleles or it will go to 1 allele. So it has a very dynamic change. And also even within the ESL population, in some cells, it will express at some time point. And when its expression becomes too much, it kicks in a feedback loop to repress itself. So it has got very variety of regulatory processes. So we study its regulation. That's the reason we have picked up nano.
Okay. So, but also at the same time, the technique that you have followed, it reminded me of the reverse genetics. So I think it is almost a reverse genetics approach using the CRISPR technology, right? If I'm correct, because because we are knowing the sequence of the gene, and then we are targeting it for understanding the function.
Yeah.
So that
CRISPRs also can be used for forward genetics.
Forward genetics. Okay. Yes. Yeah. Because you can. In that case, it can be done on any part of the genome, randomly or.
Yes. Yeah. So what has happened is people have created a lot of CRISPR libraries. So for example, there is a CRISPR library of human, mouse. So there is an array of guides, like 30,000, 40,000 guides. And with Cas9 in a single plasmid or a virus, you can infect your cells and get all sorts of knockouts and screen for your phenotypes. So the libraries are available.
But one thing I did not understand, Dr. Shekhar, you used nuclease deficient, the dead Cas9, and then you are utilizing several guide RNAs for single gene. I did not understand, when you can just block the transcription initiation, why would you require several guide RNAs for a single gene? For multiple genes, we can understand. Why would you target several aspects of the genes where you are only interested in bringing down the transcription?
Okay, that's a very good question and very important also. The reason is, these days we have very precision tools to design guides. In the beginning, there were not, one thing. Second, also, although the guides may show a very good parameters while you design them, they may not be very effective inside the cells. So as a fail-safe, we use always at least 2 guides for every target, so that we get the efficiency that we need to.
I see. Because I think after that you explained the base editors, and I think that has immense value as far as the sequence change with special. Especially for gene therapy, because there you are not invading anything, but you are just changing it without any double strand break, any break of the DNA, and you can change it. That is a wonderful way to bring about the reversal of any disease. And what I understand that this knockdown effect versus the knock-in or knockout, the knockdown effect. Are you able to score it very visibly on your line? Means, is it visible? The phenotype, is it visible?
So in many cases, yes, it is. So we as a lab don't prefer knockdowns for a simple reason. We use cells which are extremely plastic, meaning to say they are very flexible. They can change their epigenome very fast. Okay. So what happens is, when we try to do any knockdowns, which is not a genetic manipulation, the cells adopt. Initially, we see the phenotype very prominent. Later on, that is gone. So for us, it's not a great tool actually. So we use it only when we screen for things. We prefer to make knockouts, but it's a great tool for many other cell types. Or some phenotypes which don't affect the proliferation of the cells or their survival. There, I think it is pretty effective.
Yes, absolutely. Actually, for the participant, it would be also, if they can recollect, we know RNA-mediated interference, RNAi. It's actually a similar way. So you do
Yeah.
not get the complete off
Loss
of that
of
particular loss of that gene, but there is a substantial amount of repression. Yes. And of course, I think there is another question. Anuj asked, any examples where CRISPR-Cas is used in humans? Hello?
Ma'am, the question is from Anuj, I think.
Yes. Dr.
Sir, please unmute your mic, sir.
Okay, so Anuj can just allow him. He can ask.
Am I audible?
Dr. Chandrasekhar, you have to unmute yourself.
I think sir is in phone call.
Okay. So just Anuj, just wait for a while.
Okay.
Dear participants, if you have any question, then you may raise your hand or you can put your question in the chat box or in the question answer section.
Kindly participants wait till Dr. Chandrasekhar rejoins. I think he is busy at the moment. So after the session gets over, we will break for lunch, and then we will come back at 3:45. Okay. So then we will have the third session. Okay. So
Dr. P. Chandrasekhar, sir.
Yes, Dr. Chandrasekhar. Yes.
Sorry,
am I audible? No.
Yeah.
Okay. Am I audible? Okay.
Yeah, please.
So Anuj, you can, so there is a question directly from the participant. Anuj, you can unmute and ask your question.
Thank you, ma'am. Am I audible, ma'am?
Yes.
Yeah. Actually, I have a question because I have heard about these CRISPR-Cas babies. I think the news that is in the domain, that is ethical issues evolve around this. So I just want to know, is there any examples that we have used this CRISPR-Cas system in humans?
So it is allowed in humans as long as it is not germline editing. What I mean by germline editing is a mutation that is created in those cells which will take it to the next generation. So you can use CRISPRs in human cells or human organs or tissues which are somatic in nature. You can't edit a baby. So there is a moratorium throughout the world. Nobody is supposed to be editing any human embryo. Or even if an embryo is edited, it is only for the study. It can never make it beyond 14 days of human development. So that is the line that is given to all of us. We can't cross 14 days of development in human, anything that is edited. Not only that, even the embryology studies which we do in vitro can't be done beyond 14 days of human development.
Okay. So that explains. Yes. This is a very, very pertinent question.
Thank you, ma'am. Thank you, sir.
So we have another question from Vignesh. Sir, can you explain the practical approach of using the double-strand break-free gene editing technique? It would be also useful to know the different applications of this technique. So he wants you to explain the approach. I think the method. If you could explain, or you can also share some handouts, that would be more helpful, because that approach is very interesting. So what do you have to say on that, sir?
So sure. So it's actually the in case of both adenine deaminase and cytidine deaminase, if you see, these are the bacterial enzymes that are utilized. Okay, and their function is to just remove the amine group and convert it into a different analog that is read by the DNA repair system. So any, for example, any mutation that you can create, let's say UV mediated mutation, which can create such changes, will also lead to the same change, except that here we are targeting a specific base in the genome, where we are using the deaminase to change the, first remove the amine group from these bases so that it is pursued as a different base by the DNA repair system, and the corresponding change happens. So it's the same chemistry that is involved in any other mutagenesis. Okay, it is not distinct from that. And application wise, I think it has immense application potential. There is another system which I didn't talk, which also carries a correction RNA with it together. You can make multiple, it's called prime editing. You can make multiple base changes at the site, not just one base, even multiple bases at a time in one site. So that uses a different method. That uses again homologous recombination and a reverse transcriptase. But this utilizes just the deaminase, which will remove the amine group from this.
Yeah. So just one last question. So when the editing has been done in the stem cells or in the animal, when you are you how are you addressing the other off targets? Are you backcrossing it with the wild type, or how are you doing it? With general genetics, we backcross it with the wild type strain. So what is the strategy in case of this?
What we do in case of cells, there is no backcrossing, but in case of animals, yes, backcrossing.
Backcrossing, yeah. Sorry, in the cells, it's not possible. Yeah.
So in cells, what we do is, we do it for even animals. What we do is, most of these CRISPR design tools have the ability to give you the predicted off targets. So we pick up the top 5 off targets predicted for that every guide. We go and amplify those regions and sequence those regions to show that there is no off target. So in whichever cells we get such a change, we don't use those cells.
So that is safe. Yeah. Very safe. Yeah, in that case, very safe. So that really that was a really wonderful session, Dr. Chandrasekhar. It was really I really enjoyed it. And I think our participants have also gathered enough idea about the approach, which was almost like teaching from at the lab level. Today, what this particular session had actually embarked upon the strategy. So I think there are no more questions, and I really thank Dr. Chandrasekhar for his valuable time. I will also appreciate, if possible, you can share your presentation, and if possible, the strategies that you practice in your lab, some handouts. It would be really helpful for our faculties or the students sometimes later, so that I can take over and share it with them. Whichever form you feel is possible, I will appreciate that you can share it with me, and I will take it over to the participants.
Sure. I will be very happy.
Thank you so much. Thank you very much. And with this, and I again thank on behalf of the university for this wonderful session. And we will definitely be in touch with, you will be in touch with the university, you will be in touch with the participants, because now as they know you, they are going to bother you, and you will have to take all the fame. But that would be fun, actually, in a way. So this is the purpose of such a program, so that we create the link and this connection, this network, which will help for our future faculties, students to work on the CRISPR technology. So once again, thank you, sir, for your wonderful session. With this, we end the second session, and now we will break for lunch, and we'll come back at